With that, Carlson, I'll let you take over here.
All right. Thank you very much for the introduction, and thank you for the OBS team for the invitation. So today I'm going to present to you the story that we have recently published, which distinguished the lymph nodes from the spleen in generating effector T cells during a systemic chronic infection. So just very briefly, um, to introduce the topic. So in a typical acute infection, CD8 T cells are primarily in the context of antigen presentation, usually by antigen presenting cells. These cells are quickly activated, expand into effector T cells, and these cells are very robust in clearing the virus, and this allows the formation of memory cells. And memory cells basically persist long-term in the absence of antigen. They're sustained by homeostatic cytokines, and also the key features of memory T cells are the high responsiveness to the cognate antigen, to reactivation. In contrast to that, in the context of chronic infection, this differentiation trajectory does not take place. Instead, activated T cells are quickly diverted into a dysfunctional state, which we call exhaustion. Exhausted T cells are unable to completely clear the virus, although they exert considerable control to the viral replication. They're not able to completely eradicate the virus, and they coexist with the ongoing infection for long periods of time. And features of exhausted T cells, in contrast to memory T cells in effector T cells you get in acute infection, is that they persist in the presence of antigen. They exhibit decreased effector function, such as dampened cytokine production. They have low responsiveness to cognate antigen, and also they express a high level of inhibitory receptors on the surface, or so-called immune checkpoints, such as PD-1. Exhausted T cells are also commonly found in many cancers. And one of the key efforts in the field of immunotherapy is basically to target exhausted T cells by blocking these inhibitory receptors to restore their function. Just introduce the pool of exhausted T cells. So we now know that exhausted T cells are heterogeneous. There are different types of exhausted T cells, and then they can be broadly categorized into two major subsets, which we have on the left here, which we call precursors of exhausted T cells, which act as a progenitor. They express the transcription factor TCF1, and they have the ability to undergo self-renewal proliferation, as well as give rise to the cells on the right here, which are the effector exhausted T cells, which are considered the progenies. There are heterogeneity within the pool of T-PEC cells and also the TX cells. There are different models with there are actually different models of how T-PEC cells are subsetted, but in our lab, the model basically we discovered that we could segregate T-PEC cells based on the surface expression of CD62L, and we have CD62L positive T-PEC cells, which also, as Rania said, express higher level of the transcription factor MIP, which seem to also have higher capacity to self-renew and give rise to differentiate to effector cells. And there are two major types of TX cells, which one of them is characterized by the expression of CX3CR1. The other one is characterized by the lack of CX3CR1 and expression of CD101. And then we know that the CX3CR1 positive TX cells is the more effector-like subset. We now know that T-PEC cells are the targets of immune checkpoint blockade therapy, and that drives a rapid increase of effector cells via an increased expansion of the T-PEC cells. And we know that typically the clinical response in cancer ICB treatment is associated with the expansion of CX3CR1 effector-like TX cells. Therefore, it is very important to understand how this process is regulated. So just a quick recap, we know that T-PEG cells sustain chronic immunity by continuously generating T-X cells. Immune checkpoint blockade exploits the proliferative capacity of T-PEG cells, and the efficacy of the ICB therapy is dependent on a robust generation of effector TX cells. So the key question for us was basically, what are the factors that control exhausted T cell fate? And we focused on transcriptional regulation and also cell extrinsic signals. And the model we use in the lab to study exhausted T cells, one of the models that we use in the lab to study exhausted T cells, is to use the highly established systemic infection LCMV. There are different strains of LCMV. We have strains such as the clone 13 and the docile that creates a chronic infection in mice that lasts for weeks. Also, there are strains that create an acute infection that are cleared within a week very rapidly, and that drives the formation of memory T cells, while chronic infection drives the formation of exhausted T cells. Another advantage of the system is that we can actually use transgenic T cells, P14 cells that specifically recognize an epitope of LCMV, so we can actually transfer these cells adoptively to mice, infect the mice with LCMV, and then track the progress and differentiation of these transferred cells. So to look for potential transcription factors that instruct different TX cell fate, what we did was basically we did single cell RNA sequencing of P14 cells, exhausted P14 cells in the context of chronic LCMV, and what we from the spleen and the lymph node, and what we found was basically we can identify cells that exhibit T-PEG phenotype. We have 62 positive T-PEG cells here on the right, and then 62 negative T-PEG cells, and also subsets of CX3CR1 positive TX, and also CX3CR1 negative or CD101 positive TX cells. When we calculated a diffusion map and then performed slingshot trajectory analysis to try to predict how different TX cells are developed, we identified two distinct trajectories that stem from these cells here on the left, which are the T-PEG cells because they express TCF1, and we have one trajectory that corresponds to the development of CX3CR1 TX cells, and one trajectory that corresponded to the development of CD101 TX cells. And these trajectories were actually in line with other studies that have been published previously. What we identified as well was that both these two trajectories are actually marked by different gene sets. On the top, you can see that genes that are highly associated with the differentiation of CX3CR1, and on the bottom row, you can see that genes that are highly associated with the differentiation of CD101 TX cells. One of the top candidates that caught our attention that associated with the development of CX3CR1 was the transcription factor KLF2. KLF2 is a well-known important switch factor between, actually in acute infection, between circulating memory T cells and also tissue-resident memory T cells. And if we project the expression of KLF2 onto our original UMAP, we can see that expression of KLF2 were mainly focused on 62 positive T-PEG cells as well as the CX3CR1 positive TX cells, but not in the 62 negative T-PEG cells and also the CD101 TX cells. We confirmed that using flow cytometry by measuring P14 cells obtained in a chronically in a mouse infected with chronic LCMV. We can see also in the protein expression level that T-PEG cells that express CD62 also express high level of KLF2, and TX cells that express high level of CX3CR1 also express high level of KLF2. So we asked the simple question: what is the role of KLF2 in T cell exhaustion and in chronic infection? So we generated KLF2 deficient P14 cells using CRISPR-Cas9, and then we co-transfer equal number of knockout and control cells into mice. And then we infected these mice with chronic LCMV. We analyzed the immune response at the chronic time point of the infection. And one of the things that we saw immediately was that the distribution of wild type of control and knockout cells were very different in these mice. One of the striking features we've observed was that the knockout cells were very much overrepresented in the spleen and very much underrepresented in the lymph nodes. So they don't make it to the lymph nodes. But these differences in distribution do not just limit to secondary lymphoid organs. We also saw that the knockout cells were underrepresented in other organs, such as the lung and the salivary gland, but overrepresented in organs such as the liver. So how does KLF2 deficiency affect cell fate? So we address this by performing single cell RNA sequencing of the P14s from the spleen of these mice. And as you can appreciate here that they look pretty different. And we notice that in the knockout P14 compartment, there are two populations specifically that are missing that were present in the wild type. And that nicely corresponded to actually the 62 positive T-PEG cells, as well as CD3 CR1 TX cells. Instead, the KLF2 knockout populations were more enriched in cells that are corresponded that are CD101 TX cells, and also in this case CD62 negative T-PEG cells. So far we have identified that KLF2 plays an important role in putting putting cells into the lymph nodes, and also the generation of 62 positive T-PEG cells as well as CX3-CR1 TX cells. So we wanted to further explore the relationship between this concept of cells being in the lymph nodes, and also whether that has a relationship with the generation of CD62 T-PEGs and mainly the CX3-CR1 TX cells. So we wanted to test how lymph node homing and egress would affect the formation of CX3-CR1 TX cells. And for that, we picked two candidate genes that are known to be regulated by KLF2. One is CCR7, required for lymph node homing, and also S1PR1 that is required for egress from the lymph node. So we first tested the role of CCR7. We used the same technology, CRISPR-Cas9 technology, to generate CCR7 deficient P14 cells, and we co-transferred these cells with control cells, and then we did our usual chronic infection methodology. We found that in the absence of CCR7, cells are underrepresented in both the spleen and lymph nodes, indicating that CCR7 is required for cell expansion in lymphoid organs. But when we focus in the subsets of TX cells, we found that in the spleen and in the lymph nodes, in both secondary lymphoid organs, we have a selective reduction of the differentiation of CX3-CR1 positive TX cells, as you can see here. So obviously that the defect in the spleen and the lymph node was not too conclusive here in terms of whether the lymph node homing is required for the generation of CX3-CR1 TX, but at least and we think that part of the defect is coming from a difference in priming here, but at least these results were compatible with still compatible with the hypothesis that T cell homing to lymph nodes may contribute to the development of CX3-CR1 positive TX cells. And then so we decided to also look at what happens if we impair the egress from lymph nodes. So we knock out S1PR1 in P14 cells, and we transfer the cells together with control cells into the setting of chronic infection. This time, in contrast to CCR7 knockout, we have a very different picture in terms of tissue distribution. We found that S1PR1 knockout were basically trapped in the lymph node. They were overrepresented in the lymph nodes throughout infection, while there's a trend of underrepresentation within the spleen. And when we then look at the distribution of different TX subsets, we found that in the spleen, we have quite a significant reduction of cells of CX3CR1 positive TX cells. Instead, more cells are CD101 positive. However, when we look at the lymph nodes, we see the opposite trend. We see an accumulation of CX3CR1 TX cells and a reduction of CD101 TX cells. So that indicated to us that S1PR1 deficiency does not inhibit the formation of the generation of CX3CR1 per se, but then what it does is basically it traps CX3CR1 in the lymph node, and that may inhibit the accumulation of CX3CR1 in the spleen. So we basically hypothesized that, okay, we have CX3CR1 TX cells generation in the lymph nodes, and these cells then travel to the spleen. So we further show that this is a continuous process by using FTY720, which could to some degree mimics S1PR1 knockout because it targets the S1PR1 signaling pathway and it inhibits that the receptor signaling. And what we showed is that with this transient 4-day inhibition, we see a decrease of CX3CR1 TX cells in the spleen, and we saw an accumulation of these cells in the lymph nodes. And this is a quantification here, and in the same setting, we didn't see any changes in numbers in the numbers of the CX3CR1 negative TX cells, which is in line with the fact that these cells do not express S1PR1. So another recap, we show that KLF2 is required for exhausted T cell accumulation in lymph nodes during chronic infection. We show that the development of CD62 positive TPEX cells and CX3CR1 positive TX cells are completely dependent on KLF2. And we also show that the lymph node microenvironment in a KLF2 sufficient setting, the lymph node microenvironment favors the generation of CX3CR1 TX cells. So we asked the question, why would the lymph node microenvironment promote their generation? And to show you what we thought about different things. One of the ideas that we ended up testing was basically the landscape of antigen presenting cells within the difference in the landscape in APC between the spleen and the lymph node. So to characterize that, we performed single-cell RNA sequencing of the myeloid and DC compartments from the spleen and the lymph node in mice infected with chronic LCMV. We show that there are different subsets identified, including microchiip CDCs, monocyte-derived cells, CDC1, CDC2s, and also PDCs as main populations. And what we found was that in the lymph node, there's a specific enrichment, as expected, of the migratory CDCs. We confirmed that using flow cytometry. We show that there's an increase in CCR7 expression within the CDC compartment, which is typical of a typical marker of microchiip CDCs, and we confirmed that CDC1s by the expression of XCR1 from the CCR7 positive pool are mainly of migratory origin because they lack the expression of CD8 alpha, in contrast to the splenic CDC1s, which also express CCR7. Using XCR1, so CDC1 reporter mouse model in combination with reporter P14 cells, we conducted confocal microscopy of the lymph nodes, and we found indeed that TPEX cells in the lymph node paracortex within the T cell zone are found in frequent close proximity with CDC1s. This is 2 examples, and this interaction is much less likely if you move away from the T cell zone. We wanted to basically tease out the whether migratory CDCs are really driving this. So what we tried was basically we sorted different CDC1 subsets from mice infected with chronic LCMV, and we co-cultured these subsets of CDC1s with naive P14 cells and see which and try to see which subsets were able to activate the P14 cells. And what we found was basically the migratory CDC1s and not the resident CDC1s or the splenic CDC1s were capable in inducing proliferation of naive T cells in vitro. And this is the quantification here. So we basically now want to remove CDC1s from the system and see what happens. So we generated this mixed bone marrow chimera model where we combine CCR7 deficient bone marrow and also XCR1 DTR bone marrow. So basically what this mouse model allows us to do is to apply diphtheria toxin treatment and to basically deplete CDC1s with that express the DTR with the remaining CCR7 deficient CDC1s are unable to migrate into the lymph node paracortex and also the splenic white bulb. So we effectively, with the system, we could erase CDC1s in the lymph node paracortex and also the splenic white bulb. So we combine this model with P14 adoptive transfer and also chronic infection. And what we found is when we depleted CDC1s from the paracortex, we impaired TPEX cell proliferation in the lymph node, but surprisingly not in the spleen. And we also impaired formation of CX3CR1 positive TX cells in the lymph node very substantially, while this effect is much less strong, much weaker much milder in the spleen. So this tells us that CDC1 plays a critical role in the lymph node, and we show that we think that migratory CDC1s contribute at least in parts to the enhanced effector output here in the lymph node. So given that CDC1s have previously been shown to be essential for the effects of ICB, both in chronic infection and cancer, we asked the question whether the spleen and lymph node, therefore, would differ in their contribution to the overall output in response to ICB. So we conducted this simple experiment to address the question. We combine our P14 LCMV model with anti-PD-L1 treatment as the immune checkpoint blockade therapy. We gave one single dose of this treatment on day 26 after infection, and then immediately in one group of mice, we immediately blocked the egress of cells with FTY720, and we analyzed 4 days later. So in the peripheral blood, you can see that ICB or anti-PD-L1 induced a robust expansion of CX3CR1 positive TX cells into the blood. Infiltration into the blood, which was completely abrogated when we combine this treatment with FTY720. So it means that in this setting, the CX3CR1 TX cells that expanded in response to ICB could not enter the blood from the site of their generation. So we asked where are these cells then accumulated, and the answer is not in the spleen, but in the lymph node. So there is actually no observable further accumulation of CX3CR1 positive TX cells in the spleen, but then we have a very significant increase of CX3CR1 positive TX cells in the lymph node when we block egress blockade. when we block egress, indicating that the generation of these CH3CR1 cells in response to ICB, actually most of them come from the lymph node. Does it have a functional impact? We show that it does. We basically slightly adapted our FTY protocol. We now measure this time viral titer on day 30 after infection. And we show that basically in the presence of FTY720, the effect of ICB of anti-PD-L1 is reduced in certain organs significantly, and also in some organs such as the lung, is completely blocked. So this brings me to the summary. Basically, what we showed identified in this study was that you have basically two migratory patterns of exhausted T cells, one marked by the expression of KLF2, and these cells are, let's say, more favored to home to the lymph nodes, where they can encounter the immunogenic APC, such as migratory disease, as we've shown in this study. And this allows the robust formation of CH3CR1 positive progeny, and this gets disseminated via systemic circulation, and also they can enter the spleen. And in contrast, in the absence of these migratory disease, such as in the environment of the spleen, you are much less likely to produce CH3CR1 positive Treg cells. So I will finish here with this. I want to acknowledge, of course, the lab.